We're able to determine the amount of inflammation caused by cigarette smoke and RRPs by measuring the amount of inflammatory proteins that are present in an experimental system after it's been exposed. In this example, cells are exposed to cigarette smoke or aerosol from an RRP. In this video, you'll see one of our scientists carrying out an experiment to identify and measure inflammatory proteins in cells that were exposed. The cells were liquefied to produce a cell lysate, which is then analysed. This allows us to measure all the proteins that were inside the cell. Firstly, the lysate samples are prepared and diluted as necessary. The samples are then transferred into an assay plate. The experiment works on the principle of having different coloured beads, bound to which is an antibody specific to the protein of interest. The antibody will only bind to the protein it is specific for. A bead cocktail is produced in order to capture all of the proteins of interest. Up to 50 beads can be mixed together to identify and measure 50 different proteins in a given sample. Typically, we mix up to 12 beads in a given experiment. Next, the bead cocktail is added to each of the samples and left to incubate for 1 hour. The relevant proteins bind to the appropriate antibody. The excess proteins are washed off, leaving behind just the bound proteins. Next, a secondary antibody, again specific to each protein, with a tag is added to the plate. The plate is then washed and left to incubate for 30 minutes, after which it's put into the plate reader. In the plate reader, 2 lasers are used to measure the colour of the beads and the detection of the fluorescent tag. This tells us how much of each colour bead is present in the sample, therefore indicating how much protein was in the test sample. Statistical analysis allows us to determine if the levels of protein measured are upregulated, downregulated, or unchanged compared to a control experiment.